Because I have so much on my to-do list that like made me just be relaxed. Yeah.
Yeah, you can breathe again. Okay. So we're gonna finish this one first. Yes. Yes, sir. Wait, y'all. Yes. We're gonna finish 4 and we're gonna start 5. Yes. I feel like I'm missing something. I'm missing opening something. Maybe? Or maybe I got it all. I don't know. We'll find out, right? Full screen. Okay. Good morning. Hey, if you're just coming in and you're in lab, in my lab, I have graded lab reports if you want them, but only if you are in the lab and you want them. You can get them. You can get them if you want it. Here, Grace, if you want your lab report, you got yours. Okay. Who else? Philip, you want yours? And then I have Brandon and I have Kelsey. I don't think I should. Oh, okay. I was like, wait a minute, I don't have yours. Okay. All right, y'all ready? It is 10:45. Happy Friday, everybody. Yay. I was just saying to the early people that, you know, we don't really have breaks this semester. So it's really important to take the time over the weekend to like de-stress. I'm not saying don't study. I'm not saying don't do schoolwork. Do not misinterpret. It means relax a little bit, enjoy a little bit of time away from schoolwork. You know, don't burn out, okay? Don't burn out. All right. Okay, so what did the little corn say to the mama corn? Where's popcorn? It was cute, right? It was cute. Okay. So the last thing we were talking about, we were doing protein folding, right? So we have just a little bit in chapter 4 to finish, and then we're going to start on chapter 5, which is protein purification, which I love. How could you tell? How could you guess? All right. So we said that there are a couple of different reasons why proteins fold better in the cell than they do in a test tube, right? In the cell is in vivo, in the test tube is in vitro. So in vivo, they fold better. Why? What is one of the major reasons why? Concentration is one of them. The other one is chaperones, presence of chaperones. So chaperones are kind of like what their name sounds like. They help proteins fold by providing a surface for them to interact with that is beneficial for the folding process. And there are a couple of different kinds of chaperones, and we'll talk about it. But generally, you want them there as soon as the protein is forming, right? So they're going to help those newly synthesized protein strands begin to fold, right? So they're there right behind the protein making machinery. I mean, it's like they're side by side. Then you have to think that there are cellular conditions where the proteins are going to aggregate. Proteins are going to do things that they're not supposed to do. So how do you fix that? Well, you have chaperones there, and those chaperones will rescue those misfolded proteins. Do y'all know of some disorders where misfolded proteins lead to serious disorders? Alzheimer's? What is it? Not sickle cell. Yeah, prions. Yep. All those kinds of disease. Creutzfeldt Jacob. Cystic fibrosis. There are lots of them. We're going to talk about the different classifications of those in just a second, but they're very common. So really about 30% of the proteins inside of your cells have to be refolded. Not folded once, but refolded, rescued from a misfolding event. 30%. That's huge. So if you don't have that refolding going on the way it's supposed to, even if you make a normal protein, you can have a misfunctioning cell, right? Do you think a cell with tons of aggregated proteins is going to be able to do its job? No, it's filled with junk. So we have to have a mechanism that will disrupt those protein aggregates and get the cell back on track to be able to do its normal job. Oh, the thing I forgot was to put the chat up. Okay, I knew there was something, y'all. I knew there was something. Okay, there are two major classes of chaperones. There are the clamp type and there are the chamber type. So I want to talk about the chamber type first. And the chamber type, there's a little, let's see, where is it? There's an animation that talks about the two different kinds, and it's a really nice animation. So y'all should actually really check it out. It's a really nice animation. So the chamber type, what do you think of when you think about a chamber? Something that you can put it in and like close the door, right? And that's exactly how it is. It's kind of like a tube, and there's a bottom lid and a top lid. And things can go in one way, get folded, and go out. So that's a chamber type. But a clamp type, basically, it's like a clamshell. It basically clips onto the protein, helps it fold, and then releases. Those are the two major types of chaperone proteins. But regardless, this process is going to require energy, right? Think about folding a protein. If you are having to unfold and then refold something, that's going to require energy. So it's ATP dependent. So if you don't have enough ATP going on, you're going to have some problems in folding your proteins. Okay, so when we look at protein folding in general, right? Yes, you can have a chaperone to help you out, but there are protein folding events that will occur regardless of whether or not you have a chaperone present, because we do this all the time in the lab. We purify a protein or we express a protein in bacteria, right? And it will actually fold spontaneously. So how does that work? Well, if you look at how much the protein will unfold compared to temperature, what should happen as you increase temperature to a protein? Think about, okay, you're at room temperature or you're at body temperature, right? That's where proteins are normally operating. They're pretty happy. What happens if it gets hot? They start unfolding. So this is when we're going to talk about protein purification in the next chapter. Temperature is a huge deal. So if proteins get too hot, they unfold, they denature, and they're no longer functional. So if you look, as you increase the temperature, right? Do you see how it's very gradual? And then what happens? Exponential growth. What kind of a curve is this? It's an S curve or a sigmoidal curve, right? It happens very rapidly. So as soon as some things start to unfold, all the molecules unfold very quickly, right? So there's a cooperativity. And the same is true if you go in the opposite direction. If you have things at a very high temperature and you start lowering the temperature to let them renature, the same thing will happen. They'll go in this direction. You'll have all unfolded molecules, and then as you decrease the temperature, you'll hit this threshold, and they'll all start folding really rapidly. So it's a cooperativity that's occurring within the protein folding. So when one part of the molecule starts folding, oh, like that, the rest of the molecule folds. And so when we look at the protein in these states, you can actually look at the structure at different points on this curve. So if you look here, here's a partially folded protein. So that's something that's maybe over here, it's partially folded, right? It's not completely denatured, it's not completely folded, it's just partially folded. And what happens as we go this way? It folds. Do you see? This is very disordered. And you're starting to see an alpha helix here. The alpha helix gets a little bit bigger. We start to see a second green alpha helix. We get to more intermediate states. We have a red we have a green and we have a blue, 3 alpha helices. And as we progress, those alpha helices get longer and longer, and then they orient into a particular 3-dimensional shape, right? So you have basically like a nucleation event that occurs that fuels this folding process. And then the same is true if you start unfolding. If one of those things starts denaturing, it disrupts the whole thing and you go to the denatured state. Okay, so it's cooperativity in both directions. So if you think about in a cell, how does this really work? Well, there are 3 different theories on how proteins fold and how this cooperativity works. And I'm going to teach you all 3 of them. But the thing to take home about this is that all 3 happen in most proteins. It's not an all or nothing. It's not like these proteins fold this way and those proteins fold a different way. We think all 3 of them are happening in almost all proteins, with some exceptions, right? But they're all happening. Okay, so in the very first one, the hydrophobic collapse model. So what do we know about proteins? Proteins contain generally hydrophobic or hydrophilic residues, right? Tell me about those hydrophobic residues. Do they want to be sitting outside exposed to the solvent? No, but basically this is a shoestring. All the surfaces are exposed. So the first thing in the hydrophobic collapse model is that the protein strand will collapse in to protect those hydrophobic residues from interacting with the solvent. Okay, and so you get this like globular center where most of your hydrophobic residues are in there and being protected. Then once that happens, a lot of times the residues that need to be close to one another in order to make secondary structures, therefore are close together. And so then you get assembly of our secondary structures. What are our secondary structures? Alpha helix, beta strands, and then beta sheets, right? A strand is 1 arrow, a sheet is multiple together, right? Okay. Oop. I was trying to erase. Okay. So that's the hydrophobic collapse, right? And once you get that secondary structure, then that's going to feed into tertiary, quaternary, tertiary structures, all that kind of stuff. Now, the second one is the framework model. The framework model says that secondary structures will form first. Ooh, what did I do? I hit the keyboard. Okay, so in the framework model, so 2, this is the framework, you have secondary structures form first, all over the molecule, protein, whatever you want to call it, protein, right? So throughout the entire strand, you have secondary structures starting to form first. Then once those secondary structures are present, now those secondary structures can interact with one another. So I can have an alpha helix interact with a beta sheet. And that's going to give me my tertiary structures, right? Do I have an alternating alpha helix, beta strand, alpha helix, beta strand, right? Remember those motifs? Sort of, kind of, maybe. So secondary structures form first, then you get quaternary, and then you get, tertiary, and then you get quaternary, right? So that's our framework model. The nucleation model is a little bit different. The nucleation model basically says that one area within the protein folds in secondary and tertiary. So one portion, let me move this, folds secondary and tertiary. So here, do you see how this one area in the molecule, I've formed my beta sheets, my beta strands and my beta sheets, I've formed my alpha helices, and now all of these are interacting with one another. And that seeds formation of the rest of the molecule in secondary and tertiary. So this results in the rest of the molecule doing tertiary and quaternary. Do you see how the framework model is different than the nucleation? Nucleation is one little portion of the molecule seeds the rest of the molecule. Whereas the framework, it's all secondary throughout the whole molecule, then it's all tertiary throughout the whole molecule.
So the nucleation model is essentially just one part at a time.
Well, that one part forms, it's not one part at a time. It's not sequential. It's like one part forms and that seed goes outward in all directions, like a supernova, right? And it seeds the entire protein in order to fold. Does that make sense? Yeah. Okay. It's kind of cool. It's all kind of cool. Oh, I'm sorry. So this one portion folds in tertiary, secondary, results in rest of molecule, secondary, tertiary, molecule, protein, however you want to say it. Yes.
So is it fair to say, like, one differentiation you can have between 2 and 3 in the framework and nucleation is 2 is a little bit more spread out with alpha and beta?
2 is the whole protein alpha and beta, the whole protein alpha and beta before you get to tertiary, right? Before you have alpha helices interacting with beta sheets and all that kind of stuff. Yes. Yes. Yeah. I know it's kind of like, why is it important? But
It's like when you put a framework of a house, like you do framework.
right, you have to do, if you're going to frame up a house, right, what do you have to do first? You have to do all the walls before you can do the floors, or, you know, like whatever it is. You have to put a frame of the house first, then you can start filling in the gaps. Yeah, exactly. Exactly. And so all of these things really contribute to all of the proteins in the cell folding. You know, it's not one method. We don't think that the person who discovered hydrophobic collapse is right and the nucleation model is wrong. They all three happen. And that's, I think, a really good realization for science in general, that you can have multiple models that fit the same process, right? It's not one way is right and one way is wrong. Science isn't like that. And this is a beautiful example of that. Okay, watch the chaperone's animation. It's awesome. It's awesome. Okay, so I want to talk about this. Protein misfolding leading to disease. Y'all came up with some really great examples of protein misfolding. So I want to talk about a few of them, right? Let's look at this. Look at the protein structure side first, the column first. These diseases, right? Do you see how some of them cause degradation of the protein, right? Sometimes if you don't have the correct amino acid sequence, you get a mutant. You still make the protein, but then the protein doesn't hang around the way that it's supposed to, and the cell immediately degrades it. So even though you're making protein, because the cell degrades it so fast, it can't do its job. The example of that is cystic fibrosis. In cystic fibrosis, there's a pump, there's an ion pump that's required in order to get fluid off the lungs. And so there's a mutation in the gene, and the gene product is made, but then it is degraded like that. So it doesn't have a chance to pump the ions to do its job. And you get your disorder where you have all that fluid, all that mucus on the lungs, and it's very difficult to breathe, and you're way more susceptible to infections and things like that. So in all those cases, the reason that it's degraded is because you had what? A mutation in the DNA, which led to a mutant protein, right? Do you see that? All of the different ones that we talk about. Tay-Sachs is one that's actually really, really interesting. There's a Cajun heritage link to Tay-Sachs. It's really, really a terrible disease that that afflicts very small children, and it is extremely heartbreaking. And we talk about it a lot in biology and biochem 2, if you're interested in taking biochem 2. It's really interesting. Okay, so now what happens to the rest of the protein structures in these disorders? They're not degraded, they stick around, but what happens to them? They're all what? They're all aggregated. Okay, but look at this. There are two different reasons why you have, aggregated proteins. Either it's a mutant, right? Or in the case of Alzheimer's, you don't have a mutation, but your proteins still aggregate. That's a problem, right? So I think this is kind of easy to understand. If you have a mutant and the mutant changes three-dimensional shape, it changes the conformation of the protein no longer interacts with its binding partners the way it's supposed to and forms aggregations, right? That's relatively easy to get. Hey, that's Huntington's, that's how it works. But in Alzheimer's and Creutzfeldt-Jakob, there's no mutation. The protein is normal. And yet inside the cell, it aggregates. And that aggregation causes a change in the function of the cell. So what we say is that these degraded ones, these are loss of function, right? I don't make that pump. I lose the function of that pump. I'm still making the gene product, but it's not working. So it's a loss of function. But here, whether or not you have a mutant or a normal, regardless, this is a gain of function. What's the function that you're gaining? Aggregation. Is it a good function? No, but still it's a function that wasn't there before. So it's a gain of function, even though it's a bad gain of function. And it leads to a mutant phenotype. So if we look kind of at our big three examples, right? Okay, we have cystic fibrosis. Cystic fibrosis, remember we said that you have that mutation, and that mutation leads to a misfolded protein. And so that misfolded protein is degraded and you lose the function, right? You no longer make the, move the ions in the way that they're supposed to be moved. And so you end up with a disease phenotype. Huntington's disease is a mutation, and it's actually a polyglutamine track expansion. So you're supposed to have inside the gene for Huntington's a couple of glutamines in a row. But in Huntington's, they're repeated, right? It just keeps more and more and more of them. And so you can think, what's glutamine? It's charged, right? So you have interactions occurring that weren't supposed to be occurring. And so you end up with a misfolded protein. Now, this misfolded protein actually interacts with other misfolded proteins and aggregates. So this is a gain of function. So here's an example where you have a mutation where you have a gain of function. Creutzfeldt-Jakob, you actually have a normal protein being made, but there are two protein conformations. Protein conformation 1 is going to give you a functional protein. The protein is going to do its job. Protein conformation 2 is non-functional in terms of its normal job, but it gains a function in that it can now aggregate. And that aggregation is your gain of function. So there are two different ways that you can have, well, I guess technically three, but there are two different ways. You can either have a mutation to get aggregation, or you can have no mutation and you still get aggregation. So regardless, it's really, really important. Okay. Oh, and I'm not doing those. Okay. Questions about chapter 4? Yes, ma'am.
So go back to the slide where it was folding and refolding. I mean, denaturing.
Oh, this one. Whoa. This one.
Yes.
Yeah.
So whenever you denature it and then you renature it, will it go back to the exact same state it was before?
Sometimes. So your conditions have to be just right. If your salt concentration is right, if you cool it at a slow enough increment, and some proteins can and some proteins can't. So yes and no. A lot of proteins, yes. A lot of proteins, yeah. Yeah. They're pretty hardy. Proteins in general are pretty hardy. Anybody else? Good. Okay, now onto one of my other favorite subjects is protein purification, right? Now that we know how proteins function, right? We know how they fold, we know how they're made. Well, one of the biggest advances in science in the last 100 years has been the ability to produce and purify unique proteins. So think about diabetes, right? How do you treat someone who is diabetic? Depends on which one it is, right? But there are some forms that you can give them what? Insulin. So if you eat glucose, right? If you eat things that are sugar-based, basically your body needs a signal to say you can pick up the glucose, right? And so in people who are diabetic, either they don't make the insulin, or they make the insulin, but their cells are not receptive to that insulin. So to make insulin, how do we have all this insulin just lying around? How did we get it? You bought it at the store, at the pharmacy, right? How'd you get it? So we actually took bacteria and inserted the human insulin gene into bacteria. And we programmed those bacteria to make a crap ton of insulin. Now, that's great. I have a bacterial cell that is full of insulin. What do? I don't want to put bacterial other things into my diabetic patient, right? That'd be bad. That'd be bad. So what do? I'm going to purify it, right? I need to purify means, what does purify mean? It means take out the one protein that I want. What is the one protein in this bacterial cell that I want? Insulin. I want nothing else. I want it to be pure. So that's what we're going to talk about, how we do that. And that is just one example as to why protein purification is such an important advancement in science. You know, you don't think, oh, so what, you can purify a protein. No, like this is amazing. This is wonderful. So the first section we'll talk about purifying them. In the second one, we'll talk about how we can find the exact sequence of an unknown protein, or we can create a protein in the lab from scratch. It's a whole lot harder than it is to just program a bacteria to do it for us. Then we'll talk about how we can purify a protein and then actually look at the structure. You know, I've told you this is an alpha helix. I've told you these are beta strands. How do I know that? We'll talk about NMR, and we'll talk about X-ray crystallography, right? How we determine those three-dimensional characteristics. And then we'll actually talk about antibodies a little bit more, and how we can use them not only in our bodies, but also in the lab as tools, which is so insanely cool. Okay, so if you want to study proteins, right? If you study genes, we call you a what? Geneticist, right? You're studying genes. Well, if you want to study proteins, that's the field of proteomics. And you got to think about inside the cell, how many different proteins do you have? A lot. You have a lot. So think about the coding sequences in your genes. How many coding sequences do we have? About 20,000, right? Okay, so that's 20,000 different kinds of proteins. But is that the limit of diversity inside of our proteins? No, what causes more diversity on top of this 20,000 different protein products? RNA splicing. So, right, if you have a gene that has 10 exons, you could make a protein with all 10 exons, or you could make a protein with only 5, or you could make a protein with 6. And there are lots of variations, right? So that gives you quite a bit. What else? What else after it's made? post-translational modifications. Right? Oh, now we're getting more diversity, right? So we have this cell, this bagged mixture of tons of different proteins inside of it. And what I am telling you is I want you to figure out a way to study one. That's a really big, difficult job, right? So there are some people who only study one, and then there are some people who study how all the proteins interact with one another, right? So that's the proteome, right? You have the genome, you have the transcriptome, and you have the proteome. The proteome is really cool. Is the proteome going to be the same in every single cell type? No, you're not going to express all 20,000 genes, right? Depends on what cell you're in. Are you in a kidney cell? Are you in a skin cell, right? You're going to express a different set of genes, so you're going to have a different set of proteins, and you're going to have a different set of post-translational modifications. That's part of the proteome. The other part of the proteome is how those cells react to environmental cues. So the physiology, right? So what happens if you have tissue that becomes ischemic, that doesn't get enough oxygen for a long time? Like, let's say the heart tissue in a heart attack. Well, they start dying, right? But is there some way that we can maybe treat those patients to get the tissues to convert from scar tissue to back to functional tissue? Maybe if we change their proteome, right? There's all kinds of applications to this. But you can study it as a whole, or you can study individual proteins. So my background is to study individual proteins. The proteome is pretty overwhelming, and I think hard to kind of think about. But okay, so we're going to start with protein purification. And so this is the art and science of protein purification. And I like it. I like that they say this because, and they start with the art, because you can follow a procedure, right? And you can get the same result. That's science. But in order to make that procedure work, in protein purification, proteins are finicky. They just are. And so to tease out just one, you say, oh, I need to increase that salt concentration a little. Oh, I need to spend that for a little bit longer. There's little things that you have to like get a feel for. And so it's the art and science, right? So let's say we have those bacterial cells that have made insulin. So we're going to start the purification process with freshly isolated cells or tissues. So I could do this with my cells that I've expressed insulin in, or I could take a tissue sample and I want to know, I don't know, pick a protein. I want to know if this protein is present. I want to know how much of this protein is present in this tissue, right? There are all kinds of different questions that you can ask, but you're always going to start with basically live tissue or live cells. And so how in the world are you going to separate one protein from 20,000 other proteins? You have to exploit the individual characteristics of each individual protein. What does that mean? What's an example of one we've talked about in class? An individual property of a protein. What about its isoelectric focusing point? Do you remember we talked about that? That every protein has a unique set of residues, and each residue has a unique PI, isoelectric focusing point. And so if you combine them, every protein is going to have its own unique isoelectric focusing point. So that's a way for us to separate one from many, right? But it's not the only way. And we're going to talk about how we're going to exploit all the different properties, but I just want to bring up one that we've kind of talked about already. So in order to do this, we have to have a readout. We used to laugh in the lab. We used to say, people would ask, like, what do you do every day? Okay, you're a biochemist. What do you do every day? And the joke was, I mix clear liquids together. A lot of the times, that's what we did. So how do you know that all the steps that you took were successful? How do you know that you purified my protein of interest, my actin? How do I know that I got actin, right? It's a clear liquid in a tube. How do I know I did it right? I have to have some kind of a detection assay. I have to be able to detect that my product is there. So usually after the purification, so you purify first. So 1 is purify, and then 2 is test. And how do we test? We have an assay, a chemical process, right? A reaction. And if you need A plus B, and B is what you purified, right? I purified B. If I take A and add it to my sample, what should I get? C, right? If I do this and I add my sample and I don't get C, did my purification work? No, something went wrong, right? I have to have a way to analyze my results once I'm done, because you can vary one little step and at the end it all looks the same and you think you did it right, but you didn't. Been there and done that, right? Okay, so let's talk about this and give you a real life example. So y'all know fireflies, right? Their little butts glow, right? So what's in there? There's an enzyme called luciferase. And so this is in a lot of different bioluminescent organisms. So like fireflies, there's a mollusk that's actually bioluminescent. And that's actually what a lot of the studies were done on, not the firefly, but the mollusk. And it uses a substrate called D-luciferin. So if you have D-luciferin in the presence of this enzyme, luciferase, and this is the ribbon structure is luciferase, and this little substrate here is D-luciferin. So if you have these two things together, the luciferase will catalyze a reaction in the presence of ATP that will create a new product and light. So you get oxyluciferin. So you see these COOH and the OH, right? You lose those and you end up with O- on both of them, and you get light as a product. So I can try my purification scheme, then add my purified luciferase protein, and if this reaction works and I produce light, I purified my protein, right? So let's look at kind of a scheme of how that works. Okay, so I take my target organism that normally produces this luciferase and has luciferin, right? So this has the substrate and it has the enzyme. And the enzyme is also a protein, right? Right? Enzyme, protein, it's the same thing. So in this organism, if it's bioluminescent by definition, I have both the substrate and the enzyme present. Okay? So what I can do is I can take a protein, a crude protein extract, which means I take all the proteins that were inside of this organism and I have them in a test tube, right? So basically what I've done is harvested the cells and broken them open. So I have a test tube that has a conglomeration of everything that was in there. And in sample 1, if I have both the enzyme and the substrate, what should I get in this reaction? Light. I should get light. Okay, but at some point, I'm going to run out of substrate, right? The enzyme is recyclable, but is my substrate, do I have a limited, I always have a limited amount of substrate. I'm going to use it up, right? So at the end, after this, the substrate's been used up, but I still have my enzyme, okay? So I have active luciferase, and then I have my product from here, I have my oxyluciferin. Okay, that's one sample. That's what I've done to one sample. To my other sample, I'm going to heat it. What do we say happens to proteins when we heat them? They denature, they unfold. If it's unfolded, is it functional? No. Okay, so now I have inactive luciferase, but I do have the substrate. So if I mix these two samples, 1 and 2, what should happen? I should get light. Okay, now I have an assay. Now I know that I can purify, I can attempt my purification scheme. So right here, before you look at B, don't look at B. We do this. So this is like step 1, step 2. I'm going to attempt to purify. Attempt to purify luciferin, luciferase. Luciferase, right? So I've designed my assay. Now I'm going to attempt to purify luciferase. So I've purified it, and I did it 4 different ways. I did this purification scheme 4 different ways because I don't know what the best way to purify this protein is. It's a science experiment, right? It's fun. So I did it 4 different ways, but what do they all look like? Clear liquids. So what I need to do is I need to test them. So I'm going to take some of my inactive luciferase with my luciferin, and I'm going to add a little bit to each tube. See where it says add luciferin substrate? So I add a little bit to each tube. Only tube number 2 lit up. What does that tell me about tube number 2? The enzyme is present and folded correctly. So it is functional. What does that tell me about my 4 purification schemes that I tried? Only 1 worked. Hey, 1 out of 4, that's pretty good odds in the lab. You got 1 to work. Yay, that's awesome. On the supposed first try, that is, yeah, that never happens. That never happens. But this is how we design purification schemes. We think it's going to work, but we don't really know. So we try a couple of different ways, and we vary individual components of the purification scheme to find one that works. That's where like the art comes from. You know, we know what we're supposed to do. We're going to vary some things to maybe optimize, right? And maybe I had 1. Usually this is how it works. You have a couple that don't light up at all. You have 1 that lights up the best, and then you have 2 others that are like faint, right? So you kind of got it to work. Usually it's not an all or nothing sort of deal. But that's a really good example of how it works. So I said you purify, you attempt to purify the protein. How do you purify the protein? Well, you got to think of what are you starting with? A complete cell, right? Whatever organism it is, doesn't matter. You have a complete cell, and inside of that cell are all the proteins that the cell makes, everything. So the very first thing you have to do is break open the cell. And there are a couple of different ways that you can do it. But breaking open the cell and seeing the cell extract or the cell homogenate, this is all the proteins in the cell, everything. All you've done is opened it up, okay? So that means not only are all the proteins there, but they're all active, right? Theoretically. So how do you do that? Well, if you have bacterial cells, you can just kind of break them open. They're pretty easy to break open. But sometimes you got to think, well, what if I want to harvest this from the liver? What if I want to harvest this from the heart, right? That's a pretty tough tissue type. So you actually have to put some force into it in order to open them. So there are a couple of different ways that you can generate just individual membrane-bound cells, right? So you can do it through sonication. So sonication is where you send very high frequency waves at the cells and you break them open that way. You can have shearing. Watch, I'm going to show you. I'm going to come back to this slide. Don't freak out. I'm going to come back to this slide. So, right, we have our tissue, we cut it up, we put it into a tube. So sonication, you put this device into it and it emits the high intensity waves to actually open up the cell. So that's sonication, that's the first one. Shearing, so do you see how there's like a plunger that goes into that tube? So you got to think between the glass and the plunger, there's a very, very, very small gap. And that gap is actually smaller than the size of the cell. So if you force the plunger down and rotate it like this, what are you going to do? You're going to break open the cell, physically break open the cell, right? So that's shearing. Or you could treat it with mild detergents. So what do detergents interact with? The plasma membrane, the membrane of the cell, right? Because that's what detergents do. They interact with nonpolar substances, right? And what do you got in your plasma membrane? All those nonpolar fatty acid tails, right? So that's how it gets disrupted. So each method has its perks and has its drawbacks and is good for certain proteins and not for other proteins. So your protein of interest will decide which method you pick. And from that, now we have the liver cell extract or the cell homogenate. N-A-T-E, homogenate. All right. Now, I have this mixture. I'm going to go back, right? I have this mixture at this point right here. I have a mix of the entire contents of the cell. Kind of a big statement there. Is it just proteins? What else is in there? Fragments of the plasma membrane. Every single organelle, mitochondria, nucleus, right? We have everything that's in the cytosol, all the proteins and stuff that are there, right? So we have a big mix. It's not just proteins. But what we can do is we can look at the density of those different components and capitalize on their density differences using centrifugation, right? You spin it. So centrifugation will separate out the things you don't want so that we can increase purity of our target. I'm telling you, for protein purification, T-I-P-R-O-T-I-N, what is the name of the game? Playing music. What am I doing? I'm not playing music. Oh, there we go. Okay. For protein purification, what is the name of the game? What is our goal, end goal? To get only the protein of interest. So to increase the purity of our target protein. So to increase purity and concentration of protein of interest. That is a huge, important thing to remember throughout the entire process. I want more of the one thing that I want, right? The one protein that I want. Only actin. I care about nothing else, right? Whatever it is. Pick your poison. Okay. So I have this cell homogenate. Do I have high concentrations of my protein of interest? No. Here, relatively, I have low, I'm going to say protein of interest, right? Low concentration. What else do I have? Low purity, right? This is the most impure your sample can be. This is the lowest concentration your sample can be. So from here on out, those two things are going to be going up. Okay. All right. So how do we do this? And we do this in what's called cell fractionation. So we want to be able to collect different portions of this cell homogenate, right? Because you got to think, are all proteins found in the cytoplasm? No. Maybe I want a nuclear protein. So I need to find a way to isolate the nuclei. Maybe I have a mitochondrial protein that I'm really interested in. So I need to isolate the mitochondrial fraction. Or I have a cytosolic protein. So it's all, our purification scheme is all going to be dependent upon where the protein is in the cell in the beginning. So we start out and we have this mix, right? And so you take this tube and you stick it into a centrifuge And you're going to do this in lab. This is one of the experiments that we will do in lab. So pay attention now, it's going to help you later. So when you put it in, and remember, you have to perfectly balance tubes in the centrifuge. Otherwise, you're going to, it's going to wobble and it's going to create some serious damage. So anytime you ever use any centrifuge ever, you are going to balance it. Whatever weight you put on one side, look for a hole that's completely opposite and put something, whether it's a tube of water, that's the same mass, so that your centrifuge is balanced, right? I had to laugh at my physics brother who had to take a biology lab. Guess what he didn't do? He didn't balance the centrifuge, he got in big trouble. I always laugh because the physics major, come on. All right, so what happens if we put this in the centrifuge and we turn the centrifuge on? It's going to separate the components that are in there based on density, right? So the separation is based on a couple of different things. One is the component density, right? If you spin something, what happens to the heaviest things? They go to the bottom. Where are the lighter things? At the top. Now, in the cell, we don't have simply heavy things and light things, right? We have things that are a spectrum. So how can I know what's going to pellet? It's going to be dependent upon two things. And those two things are speed of my centrifuge. How many times G, right? G is gravity. How many times G am I spinning? And for how long? So duration of the spin, speed of the spin, duration of the spin, right? So if I talk about just time, as time progresses, I'll have more things being pelleted, right? So if I only spin for a short amount of time, look at from beginning to the end of the time 2. Do you see how only the white stuff is at the bottom? Everything else is more distributed throughout. If I keep spinning at the same speed, do you see the green ones have now pelleted, right? Have all come down to the bottom. So if I do a short spin, only the very heaviest things will come down. The longer I spin, the more other things that are not quite that heavy are going to pellet. Yeah. So whenever you do this, whatever spin you do, if you start and you have a mix, right? You're going to do a spin for some amount of time at some amount of speed. At the end, you're going to get a pellet and you're going to get supernatant. Now, depending on how fast, for how long, will tell you what's in the pellet versus what's in the supernatant. So I guess I should draw this, right? I guess I should do this. It's a mix, right? It's both. Okay, so let's talk about what happens when you spin at different speeds for different times. When you do that, you create what are called fractions. So if I know, let's say I know my protein is cytosolic, okay? If I know my protein is cytosolic, that means that's the lightest fraction of the entire cell. I know if I spin the first time, I'm going to pull out the heaviest things, like nuclei, right? Those big old honking nuclei. That's going to pellet first. So in that one step, I've increased my protein purification, my protein concentration, because I've pulled out those nuclei. And at every step, I'm going to pull more stuff out, and I'm going to keep increasing the purity and the concentration of my protein of interest, right? And I'm going to come back to the last step because I want you to see this. Okay, so here's my cell extract. I got everything in there. If I spin it for a low speed, 1,000 times G, this is low speed, for a short amount of time, only the biggest things are going to pellet. So I have a tube that has the pellet. So I'll do this and I'll call this the pellet. And then I have all of this, and that is called the supernatant. So if it's cytosolic, where's my protein of interest? Is it in the supernatant or is it in the pellet? It's in the supernatant. So I'm going to pour the liquid from this tube into a new tube. Now this new tube will only contain the supernatant, right? The pellet. If I want the pellet, let's say I have a nuclear protein, right? If I have a nuclear protein, I want to pour the supernatant down the drain and I want to keep that pellet. I can resuspend that pellet and now I have a nuclear fraction, right? So it all depends on where your protein of interest is located, what's your next step going to be. But let's go back to having our cytosolic protein. So we transferred our supernatant to a new tube. Now what do? We spin a little bit faster for guess what? A little bit more time. So we again have a pellet, right? And we again have a supernatant. I call it the soup, right? It's my abbreviation, the soup. So I'm going to pour my supernatant into a new tube because this is what I want to keep. But what if I was looking at a mitochondrial protein? Then what do I want to keep? The pellet, because this is the mitochondrial fraction, right? It's the next heaviest thing. But my protein's cytosolic, so I'm going to keep my supernatant. I'm going to spin it again at a higher speed for guess what? A longer amount of time. I'm going to get another what? Another pellet and another supernatant, right? So what's in the pellet? The membrane. What's in the supernatant? Just the cytosolic fraction. That's where my protein is, right? Okay, I'm going to stop right there because when we come back on Monday, we're going to talk about, okay, now that I've got all these fractions, how do I quantify that my purification scheme is working? So that'll be a great start to a Monday morning. Y'all have a great weekend. Oh, one thing, wait, before you leave. At 4 o'clock out by the back door of the main building right here, Dr. Smith is cooking gumbo. So come get a bowl of gumbo. All right, anybody on Zoom need anything?
I hit the cup, they'll serve gumbo today.
Oh, yeah?
Yeah.
No problem.
And I think,
y'all have a great weekend. I tried to invite. I keep trying. Come on. There we go.